Dan and Pamela, I'm glad brothers. Did they make it? Oh yeah, there we go. And Chris's family's here as well. It's pretty cool. And Elizabeth kind of exudes a sense of community because I know she's got current and past employers, including myself in the audience as well. So we'll get to that in a bit. So she, usually don't talk about high schools, but she graduated high school in 2010 from, as I was telling her dad, the other HB. She's from Huntington Beach. I'm from Hermosa Beach. So it was, we have the better bars. But I wasn't bringing that up for that point, but I do know that Elizabeth had a important female marine science mentor at that time that was critical in getting her involved in the career, so I just wanted to point that out. She then made her way to CSUMB, which is where she found us. Along the way, she captained the soccer team at CSUMB, which is pretty cool, and that was back when I was running the local soccer league. So we would go out, I had no idea. We would go out with a whole bunch of little kids in uniforms on the sidelines, and Elizabeth was playing. Turns out later on she'd be my own grad student. During that time, she did a lot of stuff. I think you were at IFAM at that point, as well as UROC, where she ended up coming to see me in, I think it was 2013, 2014, working with Sarah Warden, which was really cool, because at that point, although I loved it, she gave up soccer for science. Which I think was an important step because she really got to focus hard on what she was doing. She took my class in botany before she even became a Moss Lane student. So then she applied to my lab and I was like, hell yeah, I'll take you. I mean, I already knew what she could do and she was headed straight up. So we made sure that she came to Moss Lane Marine Labs in 2015, so not too long ago. During that time, she's basically gotten straight A's and she's taken every class she could possibly take. She went to Baja, she went to Chile, she took my design class, she took my botany class as an undergrad and then TA'd it later. And so she really got the most, I think, out of what you can do here at Moss Landing. Along the way, she won the Simpkins Award here and picked up some other jobs, including Center for Ocean Solutions down in Monterey, which is just really cool to see all that. And that has nothing to do with her science. That's just Elizabeth being Elizabeth, which is pretty neat, to see somebody so involved in the community doing what she's doing. Her science is kind of the fun part, though, for me, because there's, I've had a lot of grad students, and every once in a while, one of them picks up on a project that I really wanted to do. And you don't tell people that, right? But you're like, yeah, that was the one I was saving for myself. And Elizabeth picked one of those. I don't know how much is going to come out in the presentation on the background to this, but she's kind of glommed onto a topic that was raised and actually instigated, I don't want to say it badly, but incorrectly by one of the most famous phycologists in the entire Monterey Bay, Gilbert Smith, decades ago. And it's one of those questions that has been bounced around amongst established researchers for a long time, and it took Elizabeth to come forward and finally address it, which is pretty fun and pretty neat. And it's just great to see the kind of success she got with this project. So, and then I know she has some handouts here, and the decorations are great. I haven't seen tablecloths at a defense in a long time, nor an entire table full of pressings and breakfast. So it's pretty cool. Got to thank the beer pigs for making all that happen. And with that, I'm not going to go any further, but to introduce your title. So for Elizabeth Ramsay, for her master's defense, Morphological Variability within Dictyoneurum californicum and Dictyoneurum reticulatum along the wave exposure gradient on the Monterey Peninsula.
Thanks, Mike.
Thanks, Mike, for that lovely introduction. You weren't supposed to start making me cry before I talked. I'm emotional. So before I get started, I just wanted to say thank you to all of you who are actually here. I know a lot of you traveled a long ways to get here, and I'm overwhelmed with just happiness that all the people in my life have come together for this, especially since you know me. I love my seaweed, and I know you guys all are not as passionate about it as I am. So good morning, and welcome to my thesis defense. Just like any type of talk, there tends to be a general format. So first, I'm going to give you a little bit of background about why I decided to study what I did for my thesis. Then I'll introduce you to my questions, followed up by my methods. Then I will walk through my results, and then follow that up with my discussion, and then I'll finish up with some concluding notes. So to start off with some background about my thesis. So first and foremost, I'd like everyone to think back to your middle school science class, where you may have learned about Pangea, tectonic plates, and so on. You may have learned at a very young age that the world is not static, that in fact it's in a constant state of change, whether that's physical, chemical, or geological. You also learned about the theory of evolution, where the biological organisms that live within that changing world are also in a constant state of change or adaptation. So these changes or adaptations can happen over a long period of time, over multiple generations, and that would be considered genetic adaptation. This is a great example here of genetic adaptation, where the most advantageous genes are passed from generation to generation. Changes can also happen within a single generation, like you see here, where the plants sense where the sunlight is coming from, and they're triggered by that immediate environment to adjust themselves physically to photosynthesize. But these changes, however, are referred to as acclimation. So there's two different types of acclimation. The first is internal, called physiological. So I'm going to give you an example here. Maybe you have an indoor plant at home and you have it close to the window, and for whatever reason you decide to move it away from the window, and you notice that over time the leaves get darker. That's because they were triggered by losing sunlight to increase their chlorophyll A in order to continue to photosynthesize. The other type of acclimation is morphological or physical. So we'll use the same example here, where you move the plant further from the window. And now you notice that the leaf actually gets larger in size. Basically, it was triggered by its new immediate environment to increase its surface area. So acclimation, both physiological and morphological, can also happen in seaweeds. So there's four main environmental factors that tend to influence seaweeds in a physical way. There's sunlight, nutrients, temperature, and wave exposure or water motion. Water motion has been shown to be one of the most influential environmental factors in driving physical changes in seaweeds. So that's the environmental element I chose to focus on in my study. So let's take a step back for a moment. I know all of you may not be very familiar with seaweeds. So there's three main types. There's greens, reds, and browns. Within the browns, there's a type called kelp. Kelps are a favorite for morphological studies because they grow relatively fast and they tend to be quite plastic. Plastic basically means that they are highly variable based on their immediate environment and their morphological structures. So for example, the picture you see here, you're looking at two kelp blades. Believe it or not, they are the same species. The only difference is that the wider blade above was collected from a low flow environment and the blade below was collected from a high flow environment. This kelp, Acclamation arborea, is highly plastic and is influenced by water motion. So for the remainder of this talk, I just want you to remember that the small wave icon there will represent a sheltered area or a low flow environment, and the large wave icon will represent a high flow environment or a high wave exposure area. So another study done in 2007 along the New Zealand coast explored the same theory of morphological plasticity in another kelp species called Acclamation radiata. This particular coast contains many channels that feed into the ocean. So for the purpose of this study, I'm just going to give you an example of one of the channels in cartoon fashion. So water motion was one of the main environmental factors considered in this study. They followed the paradigm that was based on the distance from the mouth of the channel. So the water motion at the mouth of the channel closest to the ocean was most intense, and it gradually decreased as you moved into the channel, as you see here. So going into the study, Acclamation had two different species along the coast. However, by the end of the study, they concluded that Acclamation radiata was just morphologically plastic and was driven by the intensity of water motion within its immediate environment. So they started measuring some of the different types of morphological characteristics on the blade. So first they measured blade width, and they found that it actually statistically correlated with the water motion gradient along that channel. So specifically, blade width started off pretty skinny towards the beginning of the channel and slowly got larger as you moved into the channel. They also measured blade length, and they also found that it correlated highly with that water motion gradient in the channel. So they started off pretty short, and they eventually got longer as you moved into the channel. So for years, the seaweed that makes beautiful underwater kelp forests, Macrocystis pyrifera, or giant kelp, was thought to be many different species. Some were found shallow, some were found deep, some grew rhizomatically, some grew big giant holdfasts. It wasn't until 2009 that the morphological plasticity in Macrocystis was confirmed. And this figure that you're looking at here was the smoking gun, so to speak. So the structures at the top were found in shallow depths, and the structures at the bottom were found at deeper waters. What the study demonstrated was that water motion, or wave exposure, also correlated with depth, where in shallow waters, there was a higher intensity of water motion, and in deeper waters, it was much more calm. The study concluded that Macrocystis morphology was based on their immediate environment and not on the genetics. So these last 3 studies that I just showed you were what drove me to study another type of kelp that was actually argued over for decades. So Dictyoneurum reticulatum, the 2 left pictures, and Dictyoneurum californicum on the 2 right pictures are both common in the Central Coast area. Reticulatum is found in Mexico and parts of Southern California up to Monterey and a little bit north of Monterey. In contrast, californicum has a wider range, and you'll find it more towards just above Point Conception in California, all the way through to British Columbia. The defining morphological characteristic that set these 2 taxa apart was the midrib. So you can see the midrib here by the black arrows. Reticulatum has a very distinct midrib, and californicum does not. Reticulatum is found in more sheltered areas, and in contrast, californicum is found in more wave-exposed areas. So here is an example of a low wave exposure area. And here is a pressing of 2 reticulatum individuals collected from a sheltered environment. You can see they have a distinct midrib pointed out by the black arrows. And here is a high wave exposure area where, regardless of season, the location experiences higher wave action on average annually. And here is a pressing of californicum that I collected from a high wave exposure site. So far you've seen 2 very different kelps from 2 very different environments. So it would be a fair conclusion to say they are 2 different species. However, once I started investigating, I started finding individuals like this. They have a distinct midrib, pointed out by those black arrows, but they didn't look like either one of the 2 species I'd just shown you. And they became a lot more common as I continued to collect, which was kind of odd, totally messed up my data collection. But that's okay, because it was interesting. And then I started finding ones like this. No distinct midrib, doesn't look like either species, but my species were the closest thing you could think to match them to. So this got me thinking, maybe I needed to go back to the books and do some more research. And sure enough, after I went back to the books, I found that both californicum and reticulatum have the ability to split, splitting through the transition zone, through the blade, and down the stipe. The transition zone here is circled in red. Basically, they are not being pulled and splitting because of the exposure around them. This is a biologically programmed thing that they all do. So with this new information in mind, I started to wonder what was going on under the surface and what was driving this variety of different individuals. And that led me to my questions. So my first question was, is there a distinct morphological difference between the 2 taxa, or does it change over a wave exposure or depth gradient? My second question was, will either morph change to the other if it's transplanted to its opposite environment? And 3, does reticulatum specifically have what we would call a true midrib? That's a lot of words, so I'm going to break it down into parts. Basically, the first question is all about observations, second is all about transplants, and the third is all about midrib cell structure. And for the remainder of this talk, those icons that you saw next to each of those questions, those will be in the top left corner of each slide when I'm talking about that particular question. So keep that in mind. So for the first question, I hypothesized that individuals along the wave exposure gradient would not look identical to each other. I also hypothesized that individuals growing at depth would look different from those growing in the shallows at any given site. For the second question, I hypothesized that when juveniles from the most exposed and most sheltered sites along the wave exposure gradient were transplanted to the opposite site, they would grow to look like others in their immediate environment and not like their parents. I'd like to note here that all kelp juveniles start off looking the exact same. Californicum and reticulatum specifically both have the startings of a midrib, as pointed out by those black arrows. Basically, they're just two little veins at the bottom of the blade. I also hypothesized for my second question that if I were to take juveniles from the most exposed site and most sheltered site and put them in a common environment, they would grow up looking the exact same. And for my third and final question, I hypothesized that reticulatum would not have a distinct cellular structure within its midrib that matched species that have a true midrib. So moving on to my methods. Thank you, Mina. So in order to properly explore the morphological difference of these two taxa over a wave exposure gradient, I needed to find study sites that emulated that type of gradient. Fortunately, I live in a wonderful place for that. The Monterey Peninsula has a very distinct, well-studied exposure gradient along the coast. So looking at the map, the star is where we are here at Moss Landing Marine Labs. The photo on the right, or the white box, represents the Monterey Peninsula, and the picture on the right there is the zoomed-in version of that white box. So the colorful map you're looking at here is a wave height model from the USGS website, and this is during the fall, which I found during my study to be the most exposed time. And you can see how the colors change over that coast there, where the warmer colors have a higher wave exposure, and the colder colors have a lower wave exposure. So on average, the majority of the swell comes into the bay from the northwest, represented by the white arrow here. My most exposed site, Coral Street, was at the top there. And the rest of my sites followed the wave exposure gradient along the Monterey coast. I had a total of 5 sites. So now I'm going to move on and talk about those sites. So to tackle my first question, I headed out into the field to my 5 sites. I sampled for 4 seasons over the course of a year, collecting data from every site during each season. The first collecting season was in fall 2017, and the last collecting season was in fall 2018. During the sampling season, I would go to each site with a dive buddy, swim to about 5 feet of water, and take a heading perpendicular to the coast and drop down and follow that heading. I collected depth and growth pattern from every individual encountered on that direct heading. So from now on, I'm going to refer to growth pattern as dispersion type. There were 2 dispersion types during this study, solitary individuals and clumped individuals. Every other individual that was encountered along that heading was also collected and brought back to the lab for further analysis. And I would stop collecting at each site once I got to a depth where I no longer could find either taxa. So once I was back at the lab, I would take the individuals I had collected in the field, at least 20 individuals from each site, and collect blade measurements, midrib presence or absence data, and split presence or absence data. Once I had finished measuring each and every individual, I would then press it to keep it for later. So over the course of my study, I did press over 550 specimens. So now moving on to my methods for my transplants. So I learned very early on in this program here at Moss Landing that you're probably going to have to befriend, possibly bribe a few engineers, an electrician maybe, possibly a chemist, in order to get where you want to go. Fortunately, I have a life partner who tends to know a little bit about everything. So he helped me with a lot of these things, including my transplant platforms. So the platform here is in the center. Basically, it was 2 concrete blocks reinforced with rebar and more concrete, make it extra heavy. Then I added a plastic plate on the top there to keep it stable. Then I connected a piece of wetsuit material to that plastic plate. And inside that wetsuit material, I had little cuts in them, and that was where I would put my little juvenile holdfasts. And that's what would keep them all into place. So I had 4 platforms at Coral Street, my most exposed site, and 4 at Breakwater Pipe, my most sheltered site. and that was the most physically taxing part of my thesis, and there are 3 people here I will mention later who helped me do that. Couldn't have done it without them. Anyways, once those platforms were in place, I would then collect juveniles from both my most exposed and most sheltered sites, measure and tag them all with friendship bracelet string, and then I would transplant them to the opposite environment. And as you can see in that far right picture, they seem to look pretty happy in their new home. So the second part of the process was to conduct a common garden experiment. Juveniles were collected from both Coral Street, my most exposed site, and Breakwater Pipe, the most sheltered. They were then brought back to the aquaculture facility here at Moss Landing, where I would measure them and tag them again with that friendship bracelet string, and then throw them into the tumble culture. So the tumble culture experiment went on for 40 days, and every week I would go back to each and every individual, and I would measure its length and width, and midrib presence or absence, and split presence or absence data. And then for my third question, I collected 2 individuals that had what we would call a true midrib. Specifically, I collected Pterogophora, that picture on the far left there, Alaria in the middle there, and Undaria on the right. I also collected a few individuals of Reticulatum so that I could compare them to the true midribs. So these fresh samples were brought back to the lab, photographed and sized. I then, through each blade, cut through the blade and through the midrib in order to capture that transition zone between the blade and the midrib. I would then take those tissue samples and bring them up to the UCSC Biomed lab, and there a man named Christoph, who was very nice and willing to do this for a bottle of wine, ran them all through what we would call a microtome. Basically, it's like a meat slicer or a tissue slicer used in the biomed field, and it would just cut it small enough so that I could look at the cellular structure within the blades. So now onto the good stuff. I'm telling you, I think Rue liked my thesis more than I did. All right, so now onto my observational question first. So to remind all of you, I hypothesized that the individuals along the wave exposure gradient would not look similar to one another morphologically. And this is the visual that I observed. So what you're looking at is a compilation of pressings from collections, one from each site along the wave exposure gradient. The most sheltered site individual is on the far left, and the most exposed individual is on the far right. Notice on the most sheltered blade that it's quite wide. It also has what we would call reticulation. Basically, it's just bumps, and it's created by those veins that go up the blade. They also have a distinct midrib. But then as you move along that wave exposure gradient from left to right, notice that the distinct midrib starts to disappear. There's more splitting, and the blades become more and more thin. Interesting. Oh, so just to let you all know, for anyone who's ever tried to conduct any type of experiment ever, something always goes wrong. Nothing ever ends up perfect. So one of my sites was hard to get out to for every season for the remainder of the talk, so I'm just going to get rid of that one for now. So just remember, now I have 4 sites. Okay, so for the results for my first question, I first explored the relationship between depth and midrib. So to orient you, I have 4 sites here. Again, I have sheltered on the far left to exposed on the far right. On the x-axis is depth in meters, and on the y-axis I have midrib presence. So if an individual had a distinct midrib, I would give it a value of 1, and if it did not have a distinct midrib, I gave it a value of 0. This allowed me to run a logistic regression. Notice both my most sheltered site, the one on the far left, and the one on the far right, tend to have a very specific trend. Specifically, breakwater pipe, all the points are hovering over 1, meaning regardless of depth, they all had a distinct midrib. And the opposite trend can be seen at Coral Street, where they all hover over 0, which means none of them had a distinct midrib. So the information that I found most interesting was in the intermediate sites. Specifically, at both intermediate sites, there was a depth where the probability of finding a midrib was significant. Basically, at Lovers 2 at 4.06 meters, and at Balboa Street at 6.31 meters, and deeper, it was more likely to find an individual with a distinct midrib than without. I then explored the relationship between depth and splitting. So again, the sites are set up from sheltered to exposed. On the x-axis is depth in meters, but now on the y-axis I have split presence. So if any of the individuals had any signs of splitting, they were given a value of 1, and if they didn't show any signs of splitting, they were given a value of 0. So in this case, a lot of the sites didn't have any type of trend. However, I'd like you to focus your attention on Balboa Street here. There was a specific depth that was significantly determining when and where the splitting was occurring. So at 5.97 meters and deeper, it was more likely to find an individual not splitting, which means at this site, most of the splitting was occurring in the shallow areas. I then went and looked at the relationship between depth and dispersion type. Remember, dispersion type is either a solitary individual or a clumped individual. Again, the sites are set up sheltered to exposed. On the x-axis, again, is depth. And now on the y-axis, I have the dispersion type. So if they were found in a clump, they were given a value of 1. But if they were found solitary by themselves, they were given a value of 0. Again, the most sheltered and most exposed don't seem to have any type of trend. But Lovers 2 and Balboa Street both had a trend. So at Lovers 2 at 9.68 meters and at Balboa Street at 8.8 meters, there was that depth and deeper where it was more likely to find a solitary individual than a clumped individual. All right, so then I decided it might be interesting to look at the relationship between dispersion type and the midrib characteristic. So in this case, I ran a chi-square. So the colors on here, they're basically supposed to represent a heat map. Those values inside those cells are a percentage of the ones that were counted that had a distinct midrib to the total number counted of the clumped and of the solitary individuals. So the sites are still set up from sheltered to exposed, and then the top cells all represent the clumps, and then the bottom cells all represent the solitary individuals. I'd like to draw your attention to the dark green boxes on the left side of the screen. Notice at the most sheltered site, regardless of dispersion type, they all had a distinct midrib for the most part. And in contrast, if you were to look at Balboa Street a few cells over, notice that contrast in color. Basically, there was a significant difference between clumped individuals and solitary individuals when it came to a midrib. It was more likely for you to have a distinct midrib if you were a solitary individual than if you were a clumped individual. I also decided, well, I might as well look at dispersion type and splitting as well. So again, this is set up like a heat map. So those values inside the cells this time, instead of percent midrib, now it's percent splitting. So the percent splitting to the total number counted at each site for the clumps and the solitary individuals. So for this particular slide, I want you to focus your eyes onto the far right side of the screen. So at Coral Street, my most exposed site, notice that regardless of dispersion type for the most part, you probably were splitting. However, if you look one cell over at Balboa Street, there's definitely a significant divide. Meaning, at Balboa Street, if you were part of a clump, you probably were splitting. But if you were a solitary individual, you probably were not splitting. So now moving on to my results from my transplants. So all individuals, regardless of where they came from, went into the experiment with signs of a midrib and no signs of splitting. And again, that's those little veins going up the blade there. And when they came out of the experiment, 40 days later, still had a midrib and no splitting, all of them. You can see an example of this in the 4 pictures on the right. The top 2 are the before and after individual from Coral Street, my most exposed site, and the bottom 2 are the before and after of the individual from the breakwater pipe. The average growth rates of individuals are here in the bar graph. You'll see that on the x-axis is site collected from, so Coral Street is in darker green and breakwater pipe is in lighter green. On the y-axis is growth rate, centimeters per day. And I'd like you to notice that even though it's not quite significant, it might as well be. There's definitely a significant difference between growth rates, whether they came from Coral Street or breakwater pipe. So to make sure I ran that experiment long enough, I also collected, measured, and pressed juveniles from the field during that same month of the Common Garden experiment. The catch, though, was that all of those measured and pressed were already splitting in the field. You can see what I mean here by that black arrow, where the juvenile is actually splitting at a pretty small size. The natural individual from Coral Street there was splitting at a length of 10.5 centimeters and a width of 7.2 centimeters. And the graph you see here is the mean length and mean width, so mean length on the left, mean width on the right, of both the natural and experimental juveniles. So, and centimeters on the y-axis. Basically, I just want you to notice that there is a significant difference between both the natural and experimental juveniles of mean length and mean width. Notice that both bars from the experimental, the Common Garden experiment were significantly larger than those in the natural experiment, which means if they were in the field, they would have maybe been splitting already. So now moving on to my discussion. Fun fact though, before I move on, this pressing was made by my personal hero, Isabella Abbott, in 1970. This is a progression of reticulatum from juvenile to adult, and she actually collected all of these off of the Monterey Harbor. All right, so to remind you all again what I hypothesized, I have them in cartoon fashion here for you. This will be the general format of my discussion. So starting off with my observational question. So currently, today, Dictyoneurum reticulatum and Dictyoneurum californicum are considered two different species, and it's based off of the morphological characteristic of the presence or absence of a midrib. However, my study shows that reticulatum and californicum are actually the exact same, and their morphological differences are just based off of the immediate environment and the water motion within that immediate environment. So let me break that down for you. So when it comes to the wave exposure on that gradient there, what's accepted currently is that there's two distinct morphologies, and they keep those distinct morphologies along the wave exposure gradient. However, my study shows that in fact that's not true, that the morphological characteristics actually correlate quite tightly with the wave exposure gradient. Same can be seen by depth. So here, what's accepted currently is that there's two distinct morphologies. They may overlap in depths, but they keep that distinct morphology. However, my study suggests that in fact there's also a gradient of morphological characteristics along that depth gradient. So in order to break this down a little bit more, I'm going to go and we're going to look at each site separately. So first and foremost, I'm going to start with my most sheltered site, Breakwater Pipe. So Breakwater is actually a common dive site for most divers who are either new or in training because it's sheltered during most parts of the year, and it's an easy entrance and exit. So at 7 feet or deeper, the adults look like the one here. Basically, they have a distinct midrib. They have a lot of reticulation, so again, that bumpiness, or you can see the veins on the blade there. And there's little to no signs of splitting. However, at Lovers 2, when the water motion is a little bit more intense, there still seems to be a lot of midribs, but now a lot of them are on the insides of the blades. The blades are still pretty wide. There tends to be quite a bit of reticulation still, but again, there is an increase in splitting. And then there was Balboa Street. So Balboa Street was one of my most insightful sites because it actually followed the paradigm that I had hypothesized. Basically, the adult here is a great representation of what you would see there. Here, the midribs are basically absent. You don't find a distinct midrib until you hit 25 to 30 feet. And there's a lot more splitting going on. Reticulation seems to decrease quite a bit, and the blades start to become a lot thinner. And then there was Coral Street. So Coral Street, most people, especially divers, have a love-hate relationship with this site because it's hard to get into most times of the year, which made it exciting for me to collect my data for my thesis. So here, regardless of depth, individuals were clumped, splitting all over with no signs of a midrib. The blades were very thin, almost smooth, with streamlined reticulation. The only midribs you would see were at depth, and it usually was a juvenile, so only 10 centimeters in length. So I know I've talked a lot about midribs, so I think it's beneficial here to actually give you an idea of what a midrib actually is. So in the picture here, those black arrows are pointing to all these different types of species that have what you would call a true midrib. So the midrib term was first coined in 1909 by Griggs to be a thickened structure resembling a vein. And then in 2006, Chris Lane discussed the midrib and through genetics suggested that the midrib was an ancestral trait, meaning that only the older macroalgae on the evolutionary tree even had a midrib. Dictyoneurum is also pretty close on the phylogenetic tree to some of these older macroalgae, but not a lot of people would say they have what you would call a true midrib. In fact, in 1925, Setchell and Gardner suggested that the midrib on the Dictyoneurum reticulatum blade was only a very slight modification of the blade. And in 2006, Chris Lane actually agreed with this statement. The other interesting characteristic within these two taxa is the tendency to split. So in fact, originally Dictyoneurum was part of the Lawsoniaceae family, and the Lawsoniaceae family is well known to be part of the splitting family. So you can see some examples of that here in the pictures. So the two on the left there are both in the Lawsoniaceae family and split multiple times within one individual. The other two pictures on the right side here are actually collections made during my study. So the one on the left is from a sheltered site, and you can see that distinct midrib, but the startings of a split. In contrast, the one on the right there was collected from a more exposed site. There's no distinct midrib, and it's not splitting once, but actually twice at the same time. Oh, whoops. So this was basically the midrib, and splitting was really all phycologists had to go off of for a long time to classify these two taxa. But recent advancements in genetics have helped us dig deeper into the story. So in 2006, Chris Lane published his study on numerous kelp taxa where he investigated their genetics. His study suggested the reclassification of multiple taxa based on their genetics. Specifically, he found that Dictyoneurum reticulatum and Dictyoneurum californicum were genetically indistinct. However, in contrast, in 2007, Kuai et al. Did a similar study investigating the genetics of multiple kelp taxa and found that Dictyoneurum californicum and Dictyoneurum reticulatum were actually genetically distinct. And he hypothesized that it's maybe possible they could reproductively create hybrids. However, most genetic work pulls from herbarium specimens, meaning specimens that were pressed a long time ago and then put somewhere to be archived for later. And I have found through my own research that most herbarium specimens are archived and are usually distinct in their morphology when they're put in the archive. This is because it's really important to identify the species that you're going to be pressing, especially if you're going to be archiving it for future research. This means that most morphological hybrids, or mysterious-looking ones, are left out of the archive. So without field research, like my own, it makes sense that it's expected, on average, to find one or more of those distinct morphologies. However, my study has shown that most likely you're going to find a really weird-looking one that doesn't have either one of those distinct morphologies. So the concern is it might be possible that some of these genetic studies that are classifying these taxa are missing the intermediate ones that could be the missing link that these two species that are very separate are actually just the same species. So, because of all of this, I decided to hypothesize what I really think is going on. So here I created a GIF using scans from pressings from all of my sites. My hypothesis is that these two taxa start as juveniles with the beginnings of a midrib. However, depending on the intensity of the water motion, like at Coral Street where there's a high intensity, the individual starts splitting at a very young age, possibly before the midrib has time to move all the way up the blade. And as it continues to grow, it continues to split over and over again until eventually that midrib just disappears. So yes, this is all just observational data from the naked eye, and I get that. So I've also got some stuff in the works for genetics. So during the winter-spring of 2018, I collected 5 random adult individuals from each of my 5 sites, and then I sent those tissues to Chris Lane in his lab in Rhode Island, and those are being kept and stored for further genetic analysis. Juvenile transplants from the field were also processed and sent to Lane's lab in Rhode Island. Our hope is that the results will reveal the genetics of these 2 taxa along a wave exposure gradient, clarifying possibly my suspicions that these 2 taxa are just morphologically plastic and not genetically distinct. We also hope that the genetics clarify the diversity of phenotypes within these 2 taxa, especially when they're transplanted to the opposite environment or in a more extreme or sheltered environment. So it's important to note here that Dictyoneurum grows into 2 different ways. It can grow reproductively, like shown here. So the sori, those dark spots on the blade circled in red, have spores and through a microscopic process create macroscopic sporophytes. This process can be advantageous in calm conditions where spores can settle relatively close to the adult and grow in the same environment successfully. Dictyoneurum can also grow asexually and possibly vegetatively. This means that instead of producing spores from the sori, they grow continually from the original individual. Basically, an individual would start off looking like this, and over the course of time, many splits would occur during growth, eventually getting to the point where the number of blades, all connected to one another through their holdfasts, have the same genetic material. This type of growth would be very ecologically beneficial in places with high water motion or high wave exposure. In contrast, if they were to try and reproduce sexually, their young would be swept away, and they wouldn't have time to settle on the rock. So by growing asexually, the species can create more of itself without putting in the extra energy to go through sexual reproduction. So a topic of concern locally for kelps is the regime shift between a healthy kelp forest and an urchin barren. An urchin barren was once a beautiful kelp forest, like the picture on the left there, but due to some external environmental or biological factors, or both, the urchin populations increased while the kelp populations decreased. Now, urchins do eat kelp, but they usually eat drifting kelp that's not connected to the individual because they live in crevices away from predators. However, when this shift happens, where the regime changes over to urchins, they're able to come out and decimate the kelp forests in their surrounding environment. The Monterey Peninsula is currently suffering from this regime shift. It started actually at Coral Street, my most exposed site, unfortunately. The first kelp to disappear, though, was the giant kelp, the one that makes those beautiful underwater forests. Fortunately for me, though, I found for months that the last kelp to disappear was actually my own study species, Dictyoneurum. This could have been because Dictyoneurum has fluorotannins, basically a polyphenol or a chemical compound that deters grazers like urchins from eating them. Having these polyphenols could be an advantage ecologically for the species, especially during this regime shift. So with all that said, I think it would be very beneficial for future studies to focus on the polyphenol levels of both species during reproduction, during stressful conditions, and see if they change at all, especially over a wave exposure gradient. I also think it'd be really interesting to see a study where reproductive material from both Reticulum and californicum are brought back to the lab and grown and cultured there to see if they can produce some viable offspring. And with that, before I wrap up my talk and allow for questions, I would like to acknowledge all the people that made this research possible. So I'm going to try really hard not to cry. Okay, so first of all, I'd like to thank my piggies. So Sarah and Sarah. Sarah Jeffries was the first person to take me out into the field. You were right, Sarah. I'm going to start crying already. Sarah Jeffries took me out into the field and showed me and taught me about seaweeds while she was out collecting when she was a TA in a seaweeds class. And that's what got me going on seaweed. And then Sarah Warden was my mentor for years. She taught me about how to run an experiment and what to do, and she did not hold my hand and it was really good that she didn't. I'd also like to thank the rest of my piggies. Dan, thanks for all of the chats about my experiments, and Anne and Sloan and Mina, and yeah, I could go on forever. You guys are all awesome. I love you guys, especially for today. Thank you so much. All my dive buddies, I know I didn't list them all, so thank you if I missed you. My lab family, so especially Amanda and June. So you guys have been there for me since the beginning. We've done all the trips together, and you guys are my family. A special shout out to June and Greg and Chris, because they're the ones that helped me do those dang transplants. Each block was 100 pounds on the surface. So that was fun. And poor Greg had some jellyfish stings along the way. I'm really sorry. I'd also like to thank Stephanie Spross. She went on my first thesis dive with me. It was a disaster. Didn't even find my species. But it was good. It got me in the water. I'd also like to thank my emotional support. So, Chris, I'll get back to you in a sec, wherever you are. But my mom and my dad, the amount of times I sent my proposal draft to my dad. Dad, does it look good enough now? Can I send it to Mike? Do you think I should send it to Mike yet? It's ridiculous. My mom for all my crying calls. Scott, my brother, came out multiple times to build a system that ended up not even being used for my thesis. But he spent hours on the phone with me, on the phone with other people, trying to make it happen, and that was beyond special. Him and Chris worked very close together on that. I'd also like to thank Marina and Larry, all the positive vibes you guys sent me throughout these last 4 years. Needed it. Tom, thanks for encouraging me to keep going when I wanted to give up. Susan, Cody, and Adam and Danny, our chosen family. Thank you guys for all of the fun trips that we took when I really needed to get out of the house. So I had a few people, I'm going to talk for a minute, you guys, sorry. So I had a lot of really amazing people who didn't even know who I was and just stepped up and did a lot of really great work for my thesis. First off, the guy, Christoph, at the biomed facility, literally just called him up and said, hey, I need help and I don't have any money to give you. Could you do this for me? He said, sure, just bring me a bottle of wine. He's been amazing. And then before I decided to give up and go try and find some more expensive way to cross-section my blades, at first I went and found somebody at Hopkins, and this guy, Chris, spent hours with me cross-sectioning blades and dyeing blades, and it was just so sweet of him to do that. He had no idea who I was. I'm not even a Hopkins student. It was great. Okay, so I have to have a shout-out for him. So Ben's dad. Ben is my youngest brother's friend from college. Somehow, through the grapevine, Ben's dad, Tom, heard about my thesis and how I needed a circuitry board for my little system that Chris and Scott were building for me. And we all didn't know how to build it. And so he said, oh, I can help you with that. He built a whole circuitry board and sent it overnight to us because he was a retired electrician. I was just blown away, even drawings and explanations of how to use it properly, because he knew that we couldn't use it properly without directions. It was so great. Monterey Bay kayaks, all of the things that I used, kayaks and all, especially the kayak we cut in half. I needed that hard plastic, so thank you guys. My COS family, so Center for Ocean Solutions, I worked there for the majority of my thesis, and I had the most inspiring people surrounding me the entire time. They were so supportive throughout my entire thesis, especially my boss, Nicole. Thank you for everything. It was always I got something, my experiment failed. It's fine. Don't worry about it. We'll do the other thing, the project tomorrow, so I could go and fix my stuff. What other job has that opportunity for you? And all of the inspiring scientists, Colin, all of the women that I worked with, they were all incredibly inspiring. And it was great to have that support. My committee, okay, this is where I definitely am going to cry. So Mike, I think you know me better than I know myself in a lot of ways. You made me struggle. But I wouldn't be the confident scientist that I am today without you, so thank you. God. Tom, thank you. The amount of times that I came into your office, I know it wasn't often recently, but physical oceanography, your class was really difficult, but you were one of the most amazing teachers, and you were always there and so supportive, and I was really grateful to have you on my committee. Chris Lane, I know you're listening. I'm really excited to continue working with you. Excited to jump into the genetics part of my thesis, so get ready. Kathy Ann Miller was the one that originally discussed this idea of Dictyoneurum with me, so I had to give her a shout out. Tara Navano, where are you? Where's Tara? All of the crying afternoons in your office, thank you. You and Ivano, man, never enough hugs from you guys. The dive program, wouldn't have done any of this without the dive program. Mike, your family, I mean, they're our family too. I love them. Love your kids and your wife. Okay. And my funding from Simpkins family, I really didn't need a lot of funding, but the funding that they did provide was all that I needed. It was great. It was perfect. All right. Crap. Hey. So no words can explain how grateful I am for him. He sacrificed so many weekends over the last 4 years to help me build things, to make me laugh, to get me out of the house when I was freaking out, or coming up with ways to fix things when we both didn't know how to build something. I mean, this guy, he built me not one, not two, but 3 different sized, custom-made pressings so that I could go out and press anything I wanted in the field anytime. So supportive. All of my meltdowns at 2 a.m. because an experiment didn't work or my computer froze, whatever it was. I could go on and on. There's not enough words, sweetheart, but wouldn't have made it through it this last 4 years without you. And with that, please ask me questions. Nope, I'm ready.
Time for questions. Just to remind you, after questions are done, the committee is going to take Elizabeth into the room while you guys prepare for breakfast. You need tissues, just hand the tissues around.
Yeah, I'm good.
Any questions for Elizabeth?
Go ahead, Di.
That was really interesting and answers or examined questions on the species that we wondered about for years. So I'm wondering if now that you know this along this peninsula, which has a nice gradient of exposure, if they're within the range of the species, is there any other peninsula that you can think of, or geographic location, that sets up that same exposure gradient that you can now go and look for this pattern along that exposure gradient?
Possibly, I mean, I'm thinking along Point Conception there, but you know, I've been thinking about that, and I'm wondering if there's a more subtle gradient of morphological characteristics just from Point Conception to here, because the wave exposure definitely picks up along that gradient. However, I saw such a stark difference on such a small area that I'm not sure. Yeah, I'd have to think about that, whether there's another gradient that's on a small scale like that, but I would be really interested just to see if the depth with wave exposure gradient kind of like holds true along the coast from Point Conception to here. I think that's more interesting to me, personally. Yeah.
Go for it, Judy. That's really cool that you're sharing all your pressings with us. I feel like 500 pounds. I was still wondering for all of your sites and what we were talking about in class about trying to be representative, have full gradient and everything. How do you know that your 500 covers the full gradient of what you're trying to see? What's your rationale?
Testing me, it's okay. Okay, so yes, and I brought slides for that, if it will go. There we go. So I decided to put a few more out there. So this is all Coral Street. So they all tend to look pretty slim and no midrib whatsoever. And then Balboa Street, you start to see a little bit of a midrib starting to happen. That one that's circular almost was more at depth. And then you move on to Lovers, and now there's a midrib, and startings of a split. And then you go to Maccabee, and it almost looks like there's a lot of distinct midribs, almost no splitting. And then there's breakwater pipe. So yeah, pretty representative. Pretty stoked on that. Go, Mike.
All right, so you kind of set it up earlier that this gradient is muddying the waters of Kauai's study, where they basically said it was a hybrid, but there were still two distinct species. And I understand that they found genetic differences where Lane did not find genetic differences. But then at the beginning, you set this up that Reticulatum is a southern species that happens to be in Monterey, and californicum is a northern species that happens to be in Monterey, and so you're in this area of overlap.
Yeah.
In order to get through all that, you need to be able to also explain why reticulatum exists primarily in the south as a distinct entity that people see very often, and californicum exists in the north as a distinct entity. So these,
Right.
they represent Coral Street and Monterey Peninsula for you, right?
Yeah.
But it can't be explained, I mean, and they could be genetically distinct just due to geography.
Right.
Which is quite possibly why Kauai sees what they see.
Yeah.
But you haven't given a model to explain the geographic variability. You've given a model, because that's not a wave exposure gradient necessarily.
Right.
So how could you explain why we see this in the south and why we see Coral Street, let's say in British Columbia?
Well, just in general, like the exposure is going to be different down south versus up north. So that would make sense. And Point Conception is a good point of where species tend to divide. So it's possible that they both they started here and moved towards the points, and then eventually became genetically distinct in those furthest regions. That would be what I would think. I don't think it went the other way, where they both were distinct at those points and they came in and now have kind of merged. Although it could be a possibility too. But I think that's why. I think that they both started off separate, or they both started off at one point and then moved, and that's how they created that great divide.
Okay, and then a follow-up onto that is the other major part you found besides the wave exposure gradient is the depth gradient. Why would it be advantageous, either through acclimation or adaptation, or however you want to say it, to split more in shallow water, besides the stuff we talked about? What's the advantage of that, and why don't the deep ones do it? Because being able to have that explanation will help set up that part of the gradient as well.
Yeah, so my thought is that the reason why they split, they're actually being almost forced to split at a faster rate. And that they're doing that because they're being pulled on more often when they're in those wave exposure areas, because the shallower they are, they're being, basically it looks like a big, huge, Rastafarian hair, and it's flapping back and forth, right, in about 3 feet of water underneath the surface. So I think they're just getting pulled a lot more, and that's why they're splitting more often. It could also be because they're growing faster. So in that graph that I showed you where the juveniles from Coral Street in the Common Garden experiment were growing faster, it's possible that they're growing faster and splitting faster than those that are deeper or that are from a sheltered area.
So that's pretty much the exact same argument for the macrocystis story.
Yeah.
Good. All right, any other questions?
Yeah.
Could it be, this is maybe too obvious a question, but could there be a temperature reason for the difference in morphology between the temperature of the water?
Possibly. I don't think so here though, just because that gradient that I studied was on such a small scale. The temperature doesn't really change too, maybe a degree or two.
Southern California, which is going in.
Maybe, but then it wouldn't make sense for why they both would be here.
Yeah.
So I'm not sure, but that's a good idea. I mean, there's other things that still could be considered. So, yeah. Go ahead.
A couple things.
Why did I call on you? Okay, Dan, go for it.
I really like that you put the coon quotes in there.
You did like that, thank you.
Two of them in there.
I know.
Now for the hard one.
Okay.
So this is sort of in the speculation realm, but I was really drawn to both the clumping and the midrib part for breakwater. Now, regardless of whether or not it was split, you had a midrib for breakwater. So it makes me think, what is the relationship between the midrib and the meristem? Is there a, like, when it splits, is there a certain part of the meristem that only produces midrib tissue? And when it splits, you can see it splits sort of towards the middle.
Right.
Is that splitting actually damaging that only little tiny part that is producing the midrib, or what do you think?
Well, I think if it was biologically programmed to split, it wouldn't be setting itself up for failure, right? To split through the meristem and cause damage to the meristem. But, you know, that's kind of something that's kind of been bothering me lately, because if you're splitting through the meristem, then you're screwed. So are there two meristems? Are they splitting them in half? Because the blades still continue to grow, and they still continue to split. So I don't know, but it's really interesting.
So I don't know if you guys know this, but her third committee member, Chris Lane's in Rhode Island, watching this all through a live stream. And I keep getting text messages that we're stealing his questions. So throughout, I'm
Sorry, Chris.
getting poop emojis and things like that. Anyways, so I do want to cut it there because we do need to spend a little time with her while you guys start to enjoy what is being prepared for you so that we can let her out. So let's give a final round of applause.